Hello everyone, my name is Snehlata and I'm a PhD student and a PMRF fellow in the Department of Biological Sciences and Bioengineering, IIT Kanpur. Today we will start with a new chapter, that is Biomolecules, and this is the lecture 1 of the Biomolecule. So the first question is, what is biomolecule? Right. So before knowing what is biomolecule, first let's see that what a living organism is made up of. So a living organism are made up of two type of compounds, that is inorganic compound and organic compound, right? So the organisms are made up of inorganic and organic compounds. Now, what are inorganic compounds? So the inorganic compounds, these are compounds that do not contain certain type of bonds. For example, if you say all the compounds, if you have studied in the chemistry class that all the compounds are made up of different type of elements, right? But what is difference between organic and inorganic compound is that the inorganic compounds are the one that do not contain, these are the class of compounds that do not contain two bonds, that is this C bond and CH covalent bonds. So these are the bonds. If we see the structure of the inorganic compounds, we will see that in those compounds, these two bonds, that CC and CH covalent bonds, will not be present together. Okay, and such compounds are classified as inorganic compound. Now, what are the examples of inorganic compound? The inorganic compound can be the table salt, right? So the salt, because its chemical composition is NaCl, right? So it is not containing CC or CH bond, right? Secondly, we also have acids. So acids are also, there are certain acids that are inorganic compounds, such as hydrochloric acid, HCl. Apart from this, gases like CO2, carbon dioxide. It has carbon, but it does not have carbon-carbon or carbon-hydrogen bond. So these are inorganic compounds. Now, the second type of compound is organic compound. And these are most abundant compounds present on the earth. Okay. So what are organic compounds? That contain CC and CH covalent bonds, right? So organic compounds are the one that contain CC and CH covalent bonds. Now, what are the examples of organic compounds? The examples are carbohydrates, proteins, nucleic acids. So these all are what? They are organic compounds, right? So if you see at the example, what you can see that this carbohydrate, protein, nucleic acids, these are all present in our body, right? So if you talk about the definition of biomolecules, so you can say that biomolecules, it has two terms, right? Bio and molecule. So bio means biological, right? Related to living organism. And molecules. Okay, so you can write what are biomolecules? That all compounds that are synthesized or present in living organisms. That are either synthesized or present in living organisms, right? So these are called as biomolecules. But if you talk about the biomolecules, they are majorly organic compounds, because if you see the carbohydrate, protein, nucleic acids, lipids, these are synthesized by a living entity, right? So they majorly comprises of organic compounds. Okay, so we can also write all organic compounds. Instead of compound, you can also write all organic compounds, okay, that are synthesized or present in living organism. These are biomolecules. Secondly, these biomolecules, these are involved in the maintenance, and metabolic process. They are involved in the maintenance and metabolic process of living organism, right? So because we know that protein, it has its own role, carbohydrate, nucleic acid, and we'll also see in detail in later slides or lectures that all these compounds have a very important role in the maintenance and metabolic processes that are present in a living organism, right? So if you talk about the features of the biomolecules, what you can write? The first thing that you can write is that these are chemical compounds, right? Because they are made up of different chemicals or different elements. So these are chemical compounds. These are chemical compounds. Secondly, what type of chemical compounds? They are majorly organic compounds. And the third thing that we know, that they are synthesized and present in living organism. Now, so we saw that what a biomolecule is. Now, one important thing that we should know, that not only living organisms, if we talk about the non-living organism or anything that we are seeing around us, everything is made up of different type of elements, right? So if you see a earth, if you see a plant, if you see a chair, anything, it comprises of elements. And interestingly, these elements and the minerals that are present in the earth crust, so you see in anything, and a living organism, they are similar. Not exactly same, but similar. The earth also comprises of carbon, hydrogen, nitrogen, oxygen. Our body also comprises of these kind of elements. So the similar mineral and elements are present in both earth crust as well as in the living organism, but their relative abundance. Okay. So what is the difference? Their relative abundance vary. Okay. So the relative abundance of these chemicals or these elements vary from one type of thing to another type. Right. Now, what do we mean by relative abundance? So by relative abundance, what I mean is, like relative to other. So this term means relative to other. Now, what does this mean? Suppose we know that we have earth, and I said that earth is made up of carbon, hydrogen, nitrogen, oxygen. We have a plant. The plant is also made of the same thing, carbon, hydrogen, nitrogen, oxygen. So if someone asks you that which of these have high carbon content or higher carbon content, your answer will be earth, right? Because earth is larger than the tree. But if you talk about the relative abundance, so relative abundance means, suppose it has, for example, hypothetical situation, suppose it has a content of, say, 1000 kg, right, carbon. But the overall weight of the earth is, say, 1 lakh kg, right? 1 lakh kg. While in the case of tree, if you say, suppose it has just 100 kg of carbon, but its overall weight is just, say, 400 kg. So now if someone asks you that what is the relative abundance, which has the higher relative abundance of carbon, what would be your answer? Your answer will be, because if you calculate the relative abundance of earth, it will be 1000 by 1 lakh, right? So it will be 001. But in the case of plant, the relative abundance will be 0.25. So it will have higher relative abundance, right? So here we are talking about in the term of relative abundance. So the relative abundance of the earth constituent and our body, the exact components are same, but their relative abundance is different. Now, if we see the relative abundance of each of the elements that is present in the earth crust and human body, what we can see? We can see that the earth crust has a relative abundance of hydrogen of 0.14. So here the percent weight, or you can write relative abundance. Okay. You can also write relative abundance. So the hydrogen, if you see, Earth is larger, still it has relative abundance of hydrogen of 0.14. But in the case of human body, it is higher. It is 0.5, right? Similarly, we can see for all the other elements. One important thing here you can see is in the case of silicon, what you can see that in the case of silicon, the earth crust has relative abundance of 27.7, that is quite high. But in our body or the human body, it's negligible or it's not present. Okay. So from here, what you can say that if you talk about the human body, right? If you talk about the human body, which of these elements is most abundant or have high relative abundance or percentage weight? The most abundant element is, first of all, oxygen, right? The first element is oxygen. After that, carbon. Carbon is the second most relative abundant element. Then carbon. After this, what we can see? We can see nitrogen. And then nitrogen, what we have? We have calcium, right? It has 1.5. We have calcium. And then from calcium, after we have hydrogen. We have hydrogen, right? So this is the order of relative abundance of the elements in the human body. And the second important thing that you can see from this chart is what? That silicon, the one element that is silicon, is present in earth crust, is present in earth crust, but it is absent in human body. It is absent in human body. So human body naturally do not contain silicon. Okay, it can come in our body through certain water contamination and through other contamination, but naturally the silicon is not present in our body. Okay, so these are two important things that we can see from this chart. So now we have seen biomolecules, what it is comprises of. Next thing that we will see that how do we analyze the composition of these biomolecules. So how to analyze the chemical composition. So if you want to analyze the chemical composition of anything, say a cell or a tissue, animal tissue or a plant leaf. So first thing that what we can do, we'll take that plant leaf. If you want to analyze the chemical composition, you take a plant leaf, or say if you want to see animal tissue composition, so you will take an animal tissue. Now you want to see what are the components of which it is made up of. So for this, first of all, what you will do, you will first break it, right? It will break it or make it a paste so that everything that is present inside it come out in that paste, right? So you'll take a plant leaf or animal tissue. Now, second thing that you will do, you will make a paste or a slurry, you can say. So make a paste, you'll make a paste or slurry. And how do we do this in the case of this living organism? So what we do, we make the paste or slurry by using a thing called as mortar pestle. Called as mortar pestle. So it is like it has this kind of bowl, and then you will have something through which you will crush this tissues. Okay. So you will make a paste or slurry by using a mortar pestle, and for this purpose you will also let some solvent, right, in which it can dissolve so that the slurry can be made easily. So what you will do, and so you will use a solvent to dissolve, and the solvent is called dissolve in a solvent called trichloroacetic acid. Trichloroacetic acid. Okay. So you will take a plant leaf or an animal tissue, and then you will make it a slurry of it or a paste by using a mortar pestle and dissolve it in a trichloroacetic acid. Now, what is trichloroacetic acid? As the name suggests, it is an acid, right? And we have studied the formula of acetic acid, that is CH3COOH. So this is an acetic acid. Now, here it is saying trichloroacetic acid, right? So if you have trichloro, what you have to do? Instead of H, you have to substitute it with 3-chloro, right? You have to substitute with 3-chloro. So 3-chloro. So this will make the trichloroacetic acid. So you'll dissolve all this in the trichloroacetic acid. Now, next thing what you will do, you will pass this mixture through a filter. Okay, you'll pass this mixture through a filter, and there will be something to collect this filter. Now this is a funnel, right? In this funnel you cannot pass directly, so you will need something through it gets filtered, because the slurry will be really thin, or it will be a water kind of, so you cannot pass it directly. So you will use a different filter attached to it, and this is either made up of cheese cotton. It is either made up of cheese cloth. Sorry. So it should be made up of either cheese cloth or a cotton cloth. Okay. So the filter will be of cheese cloth or cotton cloth. Using this, you will make a filter. This is that filter. And then you will pour the slurry in it. Pour the slurry. Now, when you pour the slurry through this, you will see two type of things. The first is, some of these slurry will pass through the filter. Okay, they will pass through the filter and will be collected in the beaker or the vessel that you have kept below it. And whatever that is passed is known as filterate. Whatever that is passed is known as filterate. So what is filterate? Filterate is passed through the filter. Passed through the filter. Okay. So these are filterates. So some things will be passed through the filter. Now, there will be certain things that will be retained in this funnel or the filter, and it won't pass through it. And those things will be called as retentate. And what are these? These are the ones that are retained. These are the ones that are retained in the filter. So they have not passed through the filter. They are retained in the filter. So there will be two components, the retentate and the filterate. Okay. Now, before seeing what do we have in the retentate and the filterate, first let's see that what are the different components of which a cell is made up of. So we know this is the structure of a cell, right? And this outer membrane is a plasma membrane. Then we have cytoplasm, different organelles, everything. Now, the outer component is plasma membrane. Okay, it is a animal cell, so the outer component is plasma membrane. We know that what a plasma membrane is made up of. The plasma membrane is basically or mainly made up of lipid plus proteins, right? So the plasma membrane, it is made up of lipid plus proteins. Now we also have another component, it is called cytoplasm. We have cytoplasm. Now, if we talk about the cytoplasm, what are the all components that are present in the cytoplasm? So a cytoplasm is a place for all the chemical reactions, right? So the cytoplasm will contain most of the things that a cell has, such as it will have protein, because protein is synthesized in the cytoplasm. Now, this protein is synthesized using amino acids, right? So the cytoplasm will also have amino acids, right? It will also have amino acids. Apart from this, we have RNA from which this protein is synthesized, and this will also be present in the cytoplasm. So we can also write nucleic acid, because RNA is a type of nucleic acid. So we also have nucleic acid. Apart from this, we have water, right? We also have water. We have ions like sodium ion, potassium ion, different types of ions that are present in the cytoplasm. So this is the content of the cytoplasm, and these are the content of plasma membrane. If we talk about any other organelle as well, this organelle, because they have this membrane bound structure, they will also have lipid and proteins, right? So this is what a cell comprises of. And also when you will crush a plant leaf or animal tissue, because everything is made up of cell, right? So these are the components that will come in the slurry. This cell components will come in the slurry. And then when this slurry will pass through this filter, we will obtain two portions. The first one is called as retentate, that will be retained in the filter, and the second is called as filtrate, that will be passed through the filter. Now the question is, what will be retained in the filter and what will pass? So based on this, basically we will have two pools. Okay, we will have two pools. The first is called as acid soluble pool and the second is called as acid insoluble pool. Now, what is acid soluble pool? So the acid soluble pool will be one that will be solubilized in the acid, because here what we have used? We have used trichloroacetic acid. So the one that will be soluble in the acid will pass through the filtrate, right? And that is not soluble or that do not dissolve in the acid, that will remain on the top of the filter, right? So if we ask that what do we have in the acid soluble pool, we can write that the filtrate. First thing, the acid soluble pool will be present in the acid soluble pool will be present in the it will be present in the filtrate, right? It will be present in the filtrate. And the acid insoluble pool, it will be present in the retentate, right? It will be present in the retentate. Okay. Now the second thing, what this acid soluble pool comprises of? So it mainly comprises of the monomers, or the things that are simpler, right, or small sub molecules. So they basically comprise of monomers or simple compounds. We know what a monomer is, right? Monomer are matlab, these are single entities or basic units, you can say. So it comprises of monomers or simple compounds. But if you talk about the acid insoluble pool, so what do we have in acid insoluble pool? We will have polymers. We will have polymers or complex compounds. Polymers or complex compounds. So these are the things that are present in acid insoluble pool. Now, the third thing is we can also divide this acid soluble and insoluble pool on the basis of size. So the size of acid soluble pool will be less, that is 18 to 800 Dalton. The size of acid soluble pool will be 18 to 800 Dalton. But in case of acid insoluble pool, its size is greater than 800 Dalton. It means these are larger or bigger in size, right? So this class of molecules are basically whose size is between 18 to 800 Dalton. They are called as biomicromolecules. They are called as biomicromolecule, right? So the acid soluble pool mostly comprises of biomicromolecules because they have size of 18 to 800 Daltons. While in the case of acid insoluble pools, the molecules which has size greater than 800 Dalton, these are called as biomacromolecules. These are called as biomacromolecules. Okay. Now, what are the examples of biomicromolecules? The examples are simple sugars, because these are monomeric units, right? Sugars or amino acids. Amino acids, right? Nucleotides. Nucleotides. Ions, like sodium ion, potassium ion, calcium ion, etc. Right? So these are the biomicromolecules, and that will be present in the acid soluble pool, or the content that is filtered out or that pass through the filter. Okay. Now, if we talk about the acid insoluble pools, what are the examples? Examples are proteins, that are chain of amino acids, right? Carbohydrates, or you can write polysaccharides. Polysaccharides, there are chain of sugars. Nucleic acids. Nucleic acids. chain of nucleotides, right? So these are the example of etc. These are the example of acid insoluble pools. Now, if you see previous diagram, what you can see that the protein, the amino acids, if you see the ions, these are the component of cytoplasm, right? So if someone asks that based on the cell, you say that what are the acid soluble pool or acid insoluble pool. So you can write that in acid soluble pool, what are the major content? These are cytoplasmic content, right? So acid soluble pool basically consists of cytoplasmic content. Why? Because the cytoplasm consists of amino acids. It also consists of sugars, consists of ions. So the acid soluble pool majorly consists of cytoplasmic content. But if you talk about this acid insoluble pool, now the acid insoluble pool, the first thing that you will have plasma membrane, because plasma membrane do not contain simple molecules. Plasma membrane has lipid, proteins, right? These are complex molecules. So the plasma membrane content will be in acid insoluble pool. Second content that it will have cell wall content. The cell wall, because the cell wall in the case of plant, it is made up of cellulose, right? And cellulose is also a polysaccharide. So the cell wall content will be under acid insoluble pool. The third thing that will be in acid insoluble pool is also cytoplasmic content, because cytoplasm contain both these simple molecules as well as complex compounds, right? The cytoplasm contain amino acid, but it also consists of proteins. The cytoplasm contains sugar, but it also consists of polysaccharides. So the cytoplasmic content is present in both acid soluble and acid insoluble pool. So with this, we understood that what are acid soluble pools and what are acid insoluble pools and what are the components of it. Next, we will talk about there is one exception. There is one exception about this acid soluble and insoluble pool, and that exception is lipid. Okay. So the lipids come under acid insoluble pool. Okay. But if we talk about the size of lipid, okay, if we talk about the size of lipid, so the problem is that in the through the experiment, it is mentioned that lipids come under acid insoluble pool. But if you look at the size of lipid, so the size of lipid is less than 800 Dalton. Okay. The size of lipid is less than 800 Dalton. And based on this, in what pool it should come? Based on this, it should come under micromolecules, right? It should come under micromolecules. And the micromolecules are under acid insoluble pools. These are acid soluble pools. So it should come under acid soluble pool. But the experiment says that it comes under acid insoluble pool. So what is the reason? So the reason is, we know that our plasma membrane is made up of. So what is the reason? So we know that our plasma membrane is made up of lipid bilayer, right? So it has a structure something like this, arranged in the circular form. It has structure like this, right? So the cell membrane is made up of something like this. It is like this. But when you disrupt the cell membrane to analyze the component of the cell, what happens? They do not form a discrete unit or isolated unit like this. They do not form this. Okay, what they form? They form something like this. So they will aggregate and form a structure something like this. Okay, so this is called as aggregate. So upon disruption, instead of forming this isolated unit, they form this aggregation, and because of this aggregate, they become insoluble in acid. They become insoluble in acid. Okay. So in spite they have a molecular size of less than 800 Dalton, but because they form this aggregate, they are insoluble in acid. And that's why lipids come under acid insoluble pool. Okay. So this was the exception. And we saw that what is the reason behind it. Now, the next thing that we will see is another method of analyzing tissue. And this method is known as destructive analysis of tissue. So this is the second method through which we can analyze the composition of a tissue. And this is called destructive analysis. This method is also known as ASH analysis. Okay. So it is also known as ASH analysis. And this is mainly used to analyze. We studied in the first slide that organism is made of organic and inorganic compound. The organic compound we can analyze by a previous method that we saw. But if you also analyze the inorganic compound or inorganic constituents, if you want to analyze inorganic constituent of a cell, then this ASH analysis method is used. Now, what is ASH analysis method? For the ASH analysis, what we do, we take a living cell, living component or entities, say plant leaves. So we have taken a fresh plant leaves. Now, this plant leaf will have a fresh weight. To measure the weight, it will have a fresh weight. And what this fresh weight comprises of? It will have water, right? It will have water. It will have organic compounds. It will have organic compound, plus it will have inorganic compounds, right? So it will have three things. There's organic compounds, inorganic compounds, as well as water, because it is fresh weight. We have taken fresh leaf. Now you dry this leaf. What it will leads to? It will cause evaporation of water. It will evaporate water, right? Now, once it will evaporate water, what it will be called as? It will be dried leaf, right? It will be dried leaf. And this dried leaf, what it consists of? It will have, its water is evaporated. It will have organic compound plus inorganic compound, right? It will have organic compound plus inorganic compound, right? Now, next what? In order to get the inorganic compound, what we'll have to do? We'll have to burn it. And the process is known as combustion. To burn it by a process known as combustion. Now, in the combustion, what happens? In the combustion, two things are formed, CO2 plus H2. And this is formed by the combustion of the organic compounds. Okay. So all the organic compound upon combustion will be converted into carbon dioxide plus water. So what it will leave left with? The dried leaf upon combustion will form ash. Right. It will form ash. And this ash will comprise of inorganic compounds. This ash will comprise of inorganic compounds. So, with this method, the ash will be further analyzed, and upon analysis of the ash, what components or what inorganic compounds have been found are these. So, these are the inorganic compounds that are present in the cell and that can be found by using this ash analysis method. So, in this method, what we saw, there are different components such as sodium and potassium. And along with the organic compounds, these inorganic compounds also plays a very important role in regulating the function of a cell and overall body. For example, if you talk about the sodium and potassium ion, so what the function of sodium and potassium ion? They function in maintaining the action potential. So we'll see later what is action potential. So they are useful in maintaining action potential as well as in nerve impulse. Okay, so basically this, with the help of the sodium and potassium, signals are sent from one place, like one part of the body to another part of the body. Okay, now the second is calcium ion. If we talk about the calcium ion, the role of calcium ion is in muscle contraction It has multiple roles, out of which one role is muscle contraction. Okay. Thirdly, we have magnesium. So magnesium, it acts as a cofactor. What are cofactors? Cofactors are basically, these are the ions that help in the functioning of an enzyme. We will see in later slides as well when we will study about enzymes. So the cofactors play a very essential role in the functioning of an enzyme. Then next is water, that is important. Then we have NaCl, is another inorganic compound. So what does NaCl do? It is useful in osmoregulation. Osmoregulation means maintaining the osmotic pressure inside a cell. So NaCl helps in osmoregulation. This PO4, phosphate ion, sulfate ion, these are the components of buffer that maintain the pH of the body or the cells, right? So these inorganic compounds, though they are present in a very small amount, they have an essential role in regulating the function of a cell and the body. Okay. So with this, we saw that there are two methods by which the chemical composition of a cell or a living organism can be analyzed, right? Next, we will study about another topic, that is metabolites. So what are metabolites? Before moving to metabolites, let's first understand what is metabolism. So metabolism, in the cell chapter, what we discussed, that cytoplasm is a place where all the chemical reactions of a cell takes place, and those chemical reactions govern or tell a cell that what it has to do or how it has to behave, right? And these chemical reactions are known as metabolic reactions. So you can write that what is metabolism. So metabolism, it is a sum total of all the chemical reactions. It is sum total of all the chemical reactions that occur in a cell. Okay, that occur in a cell. This is known as metabolism. Okay, these chemical reactions are also called as metabolic reactions. These are also called as metabolic reactions, right? These are also called as metabolic reactions. Now, so what is metabolite? If this is metabolism, so metabolites, these are nothing but the compounds. These are compounds that are formed by these metabolic reactions, that are formed by metabolic reactions, right? Or chemical reactions in the cell. So the metabolites the compounds that are formed by these metabolic reactions are known as metabolites. And these metabolites, these can be converted, so they can convert from one form to another form. Okay, convert from one form to another form. So by using the help of these metabolic reactions, one metabolite can be converted to another form based on the requirement of the cell. Okay. Now, these metabolites are of two types. So if we talk about the types of metabolites, so the metabolites are of two types. The first is called as primary metabolite and the second is secondary metabolite. Now, what is primary metabolite? So the primary metabolite, the first is, the role is identifiable. Role is identifiable. Now, what do I mean by identifiable? It means that these metabolites have certain roles in that particular organism in which it is made. It is being made, right? And we know the role of, or what is the importance of the metabolites in that particular organism. Hence, their role is identifiable. But the secondary metabolites, their role is non-identifiable or not identifiable. It means that it is produced in the organism, but what actual role it plays in that organism, that we don't know in most of the cases. Okay. So in most of the cases, the role of secondary metabolite is not known. Hence, its role is non-identifiable. Now, what is the second difference between primary and secondary metabolite? Second is it is required for main metabolic reactions. Required for main metabolic reactions. Okay. Now, what do you mean by main metabolic reactions? The reactions that are essential for the survival of that cell or the organism. Okay. So these primary metabolites are required for main metabolic reactions. But these secondary metabolites, these are either byproduct. Okay. These are either byproduct. As we study in the chemistry, that there are certain main compounds that are formed and there are certain byproducts that are formed. So the secondary metabolites are either byproducts or main product, or it can also be main product of that reaction, but not useful for organisms. Always. It's not like always, sometimes, but in most of the time, they are not useful for the organism. Okay. So these are either byproducts or main products, but not useful for the organisms. Okay, not useful for the organisms mostly. Not always, but mostly they are not useful for the organisms. Okay. Now, because these are the primary metabolites are required for the main metabolic reactions, these are present in all types of the cells. Okay, they are present in all types of the cells. Okay, the primary metabolites are present in all types of the cells, but these secondary metabolites, because they are not very important for the organism, they are not present in all types of the cells. They are not present in all types of cells. By all type, I means like in bacteria, plant, animal, in all type of cells. But in the case of secondary metabolites, it might be possible that it is present in certain bacterial cell, but not in the animal cell, right? So by this is what I mean by all type of cell. Okay. Now, the function of these primary metabolites are, if we talk about the function of the primary metabolites are, they are involved in respiration. They are involved in photosynthesis. Lipid metabolism. Okay, lipid metabolism. So, the primary metabolites are involved in the functions like respiration, photosynthesis, lipid metabolism, which are all the main metabolic reactions. Okay, but the secondary metabolites, their function is, if we talk about the function of secondary metabolites, so the secondary metabolites, one function that is known is in the defense. For example, the bacteria produces this antibiotic resistance gene they have. So with this, they defend themselves from other, you can say, other cells or other living organisms. Apart from this, their function is not known in those organisms, but they are of economic importance for us. Okay. So although their function are not known in that particular organism in which it is produced, but it has certain economic importance that they can be used for us, but for human being. Okay. So next we will see what are the examples of certain secondary metabolites. So here are the certain examples of the secondary metabolites. So these are the metabolites whose functions are known or they are not very important for the organisms in which they are produced, but they can be or they are being used for human use. For example, the pigments. So the plants have different pigments like chlorophyll, carotenoids, anthocyanins. So chlorophyll is very important for the photosynthesis, but this carotenoids and anthocyanins, these are not very important for the plant, but these are used in the textile industry for providing the color. Okay. Second is alkaloid. It is also a secondary metabolite that is produced by the plants. And the example is morphine and codeine. So this morphine, this is used in the medicine as a painkiller. And this codeine, it is used in cough syrup. Right. So this is used by human beings for their own benefits. Similarly, there are other metabolites like carbenoids, essential oils, toxins, lectins, drugs, and polymeric substances like rubber, gums, and cellulose. So these are the examples of secondary metabolites. Okay, these are the example of secondary metabolites. Okay, now if we talk about the primary metabolites, example of if we talk about the example of primary metabolites, so what do we have? Everything that is important, that without which the cell won't survive, such as one example is glucose, right? Amino acids. These are also primary metabolites. These are sugars. So all these are primary metabolites that are very important for the main metabolic reactions of a cell. Okay. So with this, we cover this topic of metabolites and what are the types of metabolites. Now, before moving forward, let's solve certain questions. So the first question is, which element is present in the earth crust but absent in human cell? So there was one table that I showed in previous slide. So in the first option is phosphorus. So the phosphorus, it is present in the earth crust, but it is also present in human cells because the phosphorus is used to produce nucleic acid. So phosphorus is the component of nucleic acid. Okay. Now the second is sulfur. The sulfur is present in earth crust, right? It is also present in our body in amino acids. So there are two amino acids in which sulfur is present. The third option is silicon. So silicon is present in the earth crust, but it is not present in a human cell. So the correct answer is silicon. But let's see the fourth option. Fourth option is carbon. So we know that a carbon is present in the earth crust, and also all the biomolecules that we see are made up of carbon. So the carbon is present in present in organic compound, and the organic compounds are the one of which our body is majorly made up of. So the correct answer will be silicon. Now the next question is, the cytoplasmic content comes under. So we saw during the chemical analysis or the analysis of chemical composition of a living organism, what we saw that there are two contents, acid insoluble part and acid soluble part. So the cytoplasmic content comes under, the first option is acid insoluble part. So the acid insoluble part was one that was part of biomacromolecules, right? So the biomacromolecules comes under acid insoluble part, such as protein, carbohydrate. Okay. And then the second option is acid soluble part. So what does come under acid soluble part? Biomicromolecules, right? Biomicromolecules such as amino acids, sugar, ions, etc. Right? So if someone asks that what are the cytoplasmic content comes under, so the cytoplasm have protein, carbohydrate, amino acid, everything, right? So the cytoplasmic content comes under both acid soluble and acid insoluble part. Okay. So these are the questions, and these today we saw about the basic of the biomolecules. Now in the next lecture we will study the biomolecules one by one. So there are different types of biomolecules such as protein, carbohydrate, lipids, nucleic acids. So from next lecture onward we will see these components one by one. So that's all for today. Thank you.